Alright, so this is going to be a little bit longer of a video, I think, because this is a little bit more detailed about the process of cell respiration, although a lot of details are actually going to be overlooked. Specific numbers are going to be overlooked. It's really the process and the role that each of these molecules play that's going to be really, really important. So we're going to talk about cell respiration in more detail than the last video. So if you have not watched the previous video, please do that first, although I hope that you all have done that already, and that you understand it, and that you've asked me questions. So that being said, let's dive in. This follows along in some ways with pages 380 to 388 of your book. And so you can always refer back to that. You can look at other explanations. There's also different diagrams in the book there. And there are going to be sections of that that I'm going to direct you to rereading in addition to these video notes. But again, take the notes with the video. You should see some questions at the end of this video. But again, you want to make sure you understand it. So let's go into a couple of terms before we go into the process of cell respiration. And those first few terms are oxidation and reduction. And I've gone ahead and written some notes out to save myself some time. And I know you can pause these videos, so you might want to listen to the explanation, write down the notes, and then add any questions that you might have about that. So these first two terms, oxidation and reduction, we've talked about these before. We talked about these in terms of photosynthesis, especially when we were talking about following electrons down, actually down both the light-dependent and light-independent reactions. But most of the time we refer to it in terms of reduction, and we only talked about these electrons. We brought up this idea of oil rig, or this cheat sheet, this simplified way to think about it, but here we're going to talk a little bit more complexity with this. So oxidation is loss of electrons, reduction is gain of electrons. So and these are coupled processes, so as something is being reduced, it's gaining electrons. Those electrons are coming from another molecule, and that molecule is being oxidized because it's losing electrons. So as electrons move from one molecule to another, one molecule is oxidized and the other is reduced, right? So they're coupled processes. And so an example, real-world example you need to know about, and we just haven't gotten to this content yet, but we will when we're talking about macromolecules. We're going to be talking and seeing this thing called the Benedict's test. You might have used it in a Benedict solution, which you might have used in 9th grade, but you may not have. And it tests for certain kinds of sugars. And that Benedict solution contains this copper ion, right? The Cu2+, and as an ion, it's blue in color. But when it gets reduced, when it gains 2 electrons from the sugars, when it's combined with sugar, that Cu atom, no longer an isotope, shows up as red or orange and as a precipitate. So it comes out of the solution and changes color because it's getting reduced. It's gaining electrons, right? So and I know I was saying molecules before with the electrons. It's obviously atoms and ions as well. Okay, it's just about electrons moving from one thing to another. But you also need to know this definition of oxidation and reduction in terms of hydrogen atoms. So another way that we could define oxidation is the losing of hydrogen atoms, not hydrogen ions, hydrogen atoms. Because hydrogen atoms, I'm going to make a little note over here, that a hydrogen atom, if you remember from your periodic tables, a hydrogen atom is one proton, no neutrons, and one electron. So if we're losing a hydrogen atom, we're losing a proton, but we're also losing the electron. So in some ways it's the same process in terms of the electrons, but we don't see it written in terms of electrons. We see it in terms of hydrogen atoms. So when we lose a hydrogen atom, we're also losing an electron. When we gain a hydrogen atom, we're gaining an electron. And so the example here follows in this blue section here is with this molecule NAD+, right? NAD+ has this positive charge, but when it's combined with 2 protons and 2 electrons, that NAD plus is going to be reduced, right? It's gaining the hydrogen, a hydrogen atom, which has a proton and an electron, and then there's the other electron of the two gets absorbed into the bond, which leaves us with just a lone hydrogen ion behind. And another way to think about this is this right here. If you have 2 hydrogens and 2 electrons, that's the same as saying 2 hydrogen atoms. So this has a a positive 1 charge. When one electron gets bonded with NAD plus, it becomes NAD, and then you add another hydrogen atom to it, and you get NADH. But what you have left over is the other hydrogen proton, right? Because the electron went to kind of neutralize that charge. And that is, again, another form of reduction. You know this molecule in terms of photosynthesis when it's phosphorylated. We talked about phosphorylation last class. It's related to that process, and that's what that P stands for, is that phosphorus, right? So its phosphorylated form is NADPH, which is in photosynthesis, which is also being reduced. We talked about it in terms of electrons, but really it's this hydrogen that's being gained from NADP plus to NADPH plus H plus. And you can go back to your photosynthesis notes and add that detail if you like. You probably should. But there's a third type of oxidation reduction in terms of oxygen atoms. So when we gain an oxygen atom, when something gains an oxygen atom, it's oxidized. And I think you can see that in the root word there. But you got to be careful because we were talking about oxidation before. It's losing electrons, losing hydrogens, but it's gaining oxygens. And that's another way that something can be oxidized or reduced by losing that oxygen atom. And the reason for that is because oxygen, if you remember again from your periodic table, oxygen has an atomic mass unit of 16, 8 protons, 8 neutrons, and it therefore is so much larger than a hydrogen or electrons, than a hydrogen, that it has this great affinity for electrons, right? So it's going to attract electrons to it. So whenever you gain an oxygen atom, you add an oxygen atom, that oxygen atom is going to be pulling electrons from other parts of the molecule. And so it's removing those electrons from other components, and therefore it's getting oxidized. If you lose an oxygen, those electrons are being redistributed and added to other parts of the molecule because oxygen is no longer pulling on those electrons, and therefore it's reduced. So here's 2 reactions right here. I don't know if you know this kind of formula that well, but C7H15. So this is a hydrocarbon chain. So it's hydrogen and carbons, a long chain of carbons with hydrogens on either side of that carbon, and at the end of that whole molecule we have a carbon with 3 hydrogens coming off of it. Take that octane, oct means 8, right? So 7 and 1 make 8 carbons, plus half of an oxygen molecule, oxygen gas molecule, so just 1 oxygen gets turned into C7H15C. So there's our same root. The H3 gets changed a little bit, so there's still 2 hydrogens coming off, but an oxygen gets added in front of one of the oxygens to make our hydroxide group. And that's called octanol. I think you recognize this OH as an alcohol group. That's octanol. And this is done by certain kinds of bacteria, which is kind of really cool because this is a hydrocarbon alcohol. And that similar process could be optimized to make like biofuels, like ethanol. But that also happens through anaerobic respiration. This is what nitrifying bacteria do. We take this NO2- and add an oxygen to it. And interestingly, because of the electrons, its affinity for electrons, we still end up with a negative charge, but it's NO3 negative, and it doesn't lose its charge, but it does accept more electrons, and then that oxygen is attracting or concentrating those electrons because of its affinity for those electrons. Okay, so I know that's a lot, and I haven't even started about the process yet, but I'm going to be using the words reduction and oxidation throughout here. Okay, so let's go ahead and start talking about that process. Yet there's one more term to talk about, and that's phosphorylation. So we talked about this last class. Let me move this over a little bit. So phosphorylation is that process of adding a phosphate group PO4 3- to an organic molecule. Remember that organic is a carbon-containing molecule, and this activates. This is the really key thing here, right? So this, this activates a molecule by making it less stable, by adding more of these negatively charged groups to that molecule. It's making it less stable, which means that it's more likely to react. And those unstable, highly negatively charged parts of, let's say, an ATP molecule serve as binding sites, right? So what biochemists think is that this is the third bullet here, that specific sequence of amino acids, or what that means is certain parts of proteins serve as binding pockets, right? These locations on these proteins where these phosphorylated or phosphate groups bind in, so that kind of creates this this reaction, right? It's kind of think of like an enzyme in the substrate. It causes this reaction, and it changes the shape of the protein and causes the protein to do work. And that's really the key here. So the more phosphates you add, the more reactive this molecule can be. So we talked about this last time as well in the POGL, that the hydrolysis of ATP, the breakdown of ATP, is exergonic, which means that it is releasing energy, yet many of the cell processes are endergonic. So for your muscles to contract, they need to absorb energy from the environment. And so if you really kind of understand this well, you understand that ATP hydrolysis and your cell processes are coupled processes. In order for those cell processes to work efficiently and to work quickly and in a timely manner, the ATP needs to be hydrolyzed, and then that energy is captured by the cell processes. So they are coupled. They have to work together. So this is this kind of diagram here. This is the first part of cell respiration. It's called glycolysis, and it's only the first part of glycolysis really on this diagram. So lysis again means to break, right? Lysis means to break, and glyco is referring, think of it kind of like to glucose, right? It's referring to the sugar. It's referring, to the glucose. But remember from the previous video that glucose isn't the only reactant here, right? It could be, we could be talking about lipids. We could be talking about proteins even. But what really you need to make sure you actually probably should look at what those molecules look like, and that's in chapter 2. But glucose is the standard one that we're going to be talking about, but you should know that it's not the only one, right? So when ATP is broken down, or it's hydrolyzed down here, it's exergonic. It releases that energy, and that energy being released is being absorbed into this breakdown of glucose into glucose 6-phosphates, or G6P, right? So that energy is being released and then is being absorbed there. It's very similar to this process of oxidation and reduction. Something has to be removed from one process to be added into the other, and that's the same thing here with this ATP. Okay, so that being said, let's talk about cell respiration. Let me remove that layer. Okay, so what I went ahead and did was drew this process, and I'm going to narrate you through it, but we're going to go into a little bit more detail of each of these processes as soon as I run through this reaction. Let me get myself to a place where you can see most of what I've written. Alright, so there are 4 major parts of this process. I've labeled them up across the top. So first is glycolysis, then something called the link reaction. The link reaction actually just links the products of glycolysis to step 3, which is the Krebs Cycle, which is named after Dr. Krebs, who won a Nobel Prize for discovering this process. And then the last one is called the Electron Transfer chain, in which there's this process called oxidative phosphorylation. It actually happens in cell respiration as well. So now that, in photosynthesis as well. So what we're going to do is kind of talk about that process, but I highly suggest that you pause and draw your photosynthesis reactions, your light-dependent reaction and your light-independent reaction. Label it for oxidation and reduction. See if you can follow the electrons and the hydrogens and the oxygens, and make sense of that. And then you can compare those processes to this one. So what we have in glycolysis, in step 1, this is all glycolysis inside of this cell membrane, but in the cytoplasm, not in the mitochondria. That's what this red organelle is. We have glucose that actually will move across the cell membrane. Glucose is a big molecule, so it can't just move and shove its way through the phospholipid bilayer, but it needs its own glucose transporter. It goes through this protein, and for now we're just going to symbolize it as this cylinder. But glucose will move into the cell, and it goes through a series of processes that will create this pyruvate. So we'll again, I'm going to zoom in on step number 1, but what you'll see is that ATP is hydrolyzed so that that energy can then be absorbed into this chemical pathway, this metabolic pathway. NAD plus is going to be reduced with the gain of these hydrogens and electrons to make NADH plus hydrogen ion, and the green line will follow in a second. And it creates, in the end of this whole process of glycolysis, 2 3-carbon pyruvates. So remember that glucose is a 6-carbon molecule. It actually hydrolyzes into 2 individual 3-carbon pyruvates. I will add a little note here that anaerobic respiration, when there is no oxygen, present, this will go through fermentation. So in the previous video, I talked a little bit about fermentation in animals and yeast, and that's really what's key here. And in bacteria that don't have a mitochondria, this is really the limit of what they can do. And so that what they're making is a gross yield of 4 ATP. But they use 2 ATP to do so, and we do as well. So really the net output of ATP from glycolysis is simply 2 ATP molecules, which isn't that much, right? If you're thinking about this with the analogy of 1 ATP molecule is a $1 bill, it's not a lot of money that's coming from 1 glucose molecule. And that's what's key with this idea of fermentation, that you can still make some ATP, but it's not a lot. And this process, at least specifically in humans and yeast, is highly effective when there's an immediate need for ATP. You don't have to go through the rest of this process of all the stuff that's happening in the mitochondria to get a few ATP. So when you need a quick burst of energy, fermentation, lactic acid fermentation for you is not a bad thing. But it's not sustainable for the long run. What ends up happening with these pyruvates is that these pyruvates are able to then move through a series of protein in this membrane and this membrane into what's called the matrix of the mitochondrion. The matrix is this middle part, but I want you to notice that there are, while this cell membrane has a phospholipid bilayer, the bilayer is represented by these 2 lines, the mitochondria has a double phospholipid bilayer. So here's one phospholipid bilayer, and here's another one. And this inside one is called the intermitochondrial membrane. Inside of the intermitochondrial membrane is this thing called the matrix. Then between the 2 membranes, we have this intermembrane space and the outer mitochondrial membrane. So this pyruvate finds its way into the matrix, and the and I put like finds its way in air quotes, because there are proteins that help it move, but this pyruvate then goes through what's called the link reaction. And in the link reaction, this 3-carbon pyruvate will break and release a carbon dioxide into the atmosphere. Now, you should remember that this oxygen and CO2 was part of the pyruvate, which is was part of the glucose, right? So when we say 3-carbon pyruvate, it's not just 3 carbons, but we denote it, we follow the number of carbons to make sure that we're following the number of atoms correctly. So this 3-carbon pyruvate loses a carbon, and this acetyl-CoA enzyme is a 2-carbon molecule. That CO2 will, as a nonpolar molecule, will diffuse across this membrane, diffuse across that membrane, diffuse across this membrane, and in your body find its way to the capillaries that are streaming right to each one of your cells. That carbon dioxide goes to the hemoglobin, finds its way to your lungs, and and diffuses out of your lungs, and you exhale it out of your body. So that's the CO2. It's a byproduct. But the acetyl-CoA isn't done yet. So the acetyl-CoA will go into this process called the Krebs cycle. So I tried to draw it cyclically. This 2-carbon molecule gets bonded with a 4-carbon molecule that's coming in right here. Okay, a 4-carbon molecule. So now we end up with 6 carbons, and we lose 2 carbons through CO2, which also diffuse out of the cell. But there's a component that gets reduced. NAD plus gets reduced. Actually, there are 2 NAD pluses. They get reduced to NADPH plus H plus. And ADP gets phosphorylated to ATP. And there's a new molecule called FAD that also gets reduced to FADH2. So it's gaining hydrogen atoms. Those hydrogen atoms have a proton and an electron, right? So it's getting reduced. And these molecules, the NADH and the FADH2, and then this NADH from glycolysis, all find their way into or towards the inner mitochondrial membrane. And there are these proteins here that will pump those hydrogen ions into the intermembrane space. I hope this looks familiar to you. This is very similar to what's happening in the light-dependent reaction with the electron transfer chain, and that's what this process is, the electron transfer chain. So the hydrogens get pumped across, the electrons lower their energy as they are brought down the electron transfer chain. They're passed from protein to protein to protein. There's a high concentration of hydrogen ions being built up here. Fortunately, this intermembrane space is really, really small, so there's not a lot of volume. So this proton gradient can be built up very quickly. The hydrogen ions then are released through ATP synthase, right? So this pink protein represents ATP synthase. As hydrogen ions move through it, oxygen is going to combine with those hydrogens and the electrons that move down this electron transport chain. So the electrons, think back to your photolysis. In photolysis, water gets broken into electrons, hydrogen, or protons, and oxygen. Here, that oxygen gas gets recombined with hydrogen ions and electrons to give off water. And this is critically important. Oxygen is known as an electron acceptor. So again, we're following electrons. It's also accepting hydrogen ions, but it's called an electron acceptor. The reason that that's so important is that these hydrogen ions, if they were allowed to pass through independently without combining with oxygen gas, we would find an equilibrium of protons in the inner membrane space and the matrix. We would not be getting rid of these hydrogen ions. And by bonding with this water, we're ensuring that there's a concentration gradient from the intermembrane space to the matrix. And so, if you look back at your photosynthesis, you should know that your drawing for photosynthesis was incomplete. You might have found that some models of the process of photosynthesis are written like this. water and CO2 with the energy from light give off glucose and oxygen and water, right? So water is an electron carrier or acceptor in photosynthesis as well. It's for the same reason, to ensure that there's a concentration gradient. But this ATP synthase is doing the same process of converting ADP plus a lone phosphate into ATP. All right, so what I'm going to do, actually, this process I think you probably know well enough, but it's this process of glycolysis and the link reaction and the Krebs cycle that I want to go into a little bit more detail on. But what I guess there's one other component I should add, is that these NADH plus H plus and the FADH2s bring those hydrogens here, but then what happens to the rest of the molecule when they release those hydrogens? Well, they return back, right? So they return back here in their oxidized form, NAD+, or they return back here to NAD+, or they return here, they return here as FAD in their oxidized forms, picking up new hydrogens that are coming from the next glucose molecule. Now, most of this ATP is made here because of this concentration gradient. So only 2 ATP are made, new ATP are made through glycolysis, but on the order of 32, 34, 36 numbers of ATP are made through this component, through the ATP synthase in the electron transfer chain. So oxygen is critically important here, because without oxygen we would not be able to maintain this concentration gradient. ATP synthase would not function. It would not make lots and lots of ATP. So your cell actually doesn't go through this when there's no oxygen. Even though there's no oxygen here in Krebs cycle doesn't happen without or in the absence of oxygen, because without oxygen accepting hydrogen ions, new hydrogen ions aren't being dropped off into the inner membrane space by these carriers NADH and FADH2. So that process isn't happening. So it can't get new hydrogens from glucose. But instead, what your cells do is say, all right, we're in a holding pattern. We can't go into the mitochondria. There's nowhere to go in the mitochondria. So they're going to go through fermentation to unload some of those electrons and hydrogens into making lactic acid or ethanol, so that they can go back and get more electrons and hydrogens in the oxidized NAD+ to make just a couple more ATPs. Okay, so I'm going to add, so that was the end of what I had prepared before the video. But what we wanted to do now, I think what I want to do is talk a little bit about glycolysis first. Glycolysis. So in glycolysis, you need to make sure that you understand that this is really glucose being broken down through a series of processes. This is representative of what we would call generally metabolism. It's a series of biochemical reactions. Glucose being converted to what we talked about earlier, which is glucose 6-phosphate, which then gets converted to a fructose, which is another type of sugar. It's an isomer of glucose, so fructose 6-phosphate. Now this phosphate joined this molecule from ATP. So we don't have ADP plus P because that P got absorbed into glucose 6-phosphates. And then that fructose 6-phosphate gets turned into, well, I'm not going linearly anymore, to fructose 1,6 biphosphate. So the 6 and the 1,6 refer to which numbered sugar of the fructose the phosphates are added to. So where does the second phosphate come from? I hope you figured this out already. ATP to ADP, that third phosphate gets incorporated into this molecule. So this has 2 phosphates in it now. But we're not quite done yet. This is not pyruvate. We said that glycolysis ends in pyruvate. So this fructose biphosphate, which has 6 carbons in it, actually divides, or gets broken down in a chemical pathway into 2 triose phosphates, which is a familiar molecule to us because it's in the Calvin cycle. Triose phosphates, right? These are 3 carbon molecules. It's a 3 carbon sugar. OS means sugar. 3 carbon sugar with a single phosphate group attached to it. And then it gets, I'm just going to do this down one pathway here. It is hydrolyzed, it's oxidized into glycerate. I hope you can finish this word, glycerate 3-phosphates, or G3P, which again comes from photosynthesis. So glycerate 3-phosphate is oxidized because something is reduced, and that is the NAD plus to NADH plus a hydrogen ion. And again, if you're confused about this, go back to the beginning of this video where we talked about that this is a form of reduction. So we got this G3P molecule, which then will be converted into one more step into a molecule that we will call pyruvate, okay, or pyruvic acid, okay, pyruvate or pyruvic acid. And again, that's happening twice, right, to the other triose phosphate or the other TP. And I think, yeah, well, that I don't want to go into any more detail than that, but this pyruvate then has the cell has a quote-unquote decision to make on whether or not it's going to go to aerobic respiration or not. So we're going to follow this aerobic respiration pathway into the link reaction. So the link reaction, which is number 2, the second process, the link reaction, is this process that this pyruvate gets converted to acetyl-CoA enzyme. All right, so remember that this pyruvate is a 3-carbon molecule, and in reality we have 2 of these that were resulted from 1 glucose molecule. But that pyruvate, which is a 3-carbon molecule, gets converted into a 2-carbon molecule. I know, stunning. So in order to lose that third carbon, CO2 is removed, but there's also a reduction of NAD plus to NADH plus H plus. So that's being oxidized, and then the role of this again is to go to the electron transport chain to be used to create that hydrogen or that proton gradient to allow for oxidative phosphorylation, right? So oxidative phosphorylation is this oxidation process of phosphorylating a molecule. So ADP is phosphorylated to ATP through this oxidative process of the hydrogen ions gaining oxygen atoms, right? So H2O is the result of that oxidation process. So that's called oxidative phosphorylation. It's also happening in photosynthesis. So this acetyl-CoA enzyme is then produced at the end of this link reaction to make, to go into yet the next process, which is the Krebs cycle. There's a good diagram of this link reaction in your book on 383. It's figure 3 of the book. It's nothing major, but it's just a different kind of visual with a little bit more detail that might help you. But the third phase here is the Krebs cycle. And the Krebs cycle is a little confusing with lots of detail. We're going to kind of do an overview here. I'm going to try to do this cyclically, and there's a model of this in your book as well. Sorry about that. Let me undo that. All right, so we have this cyclical. It's the best circle I can do at 4 in the morning. Okay. So what we have is this cycle, and the reactant that's coming in here is that acetyl-CoA, right? So acetyl-CoA enzyme is a 2-carbon molecule. And the cyclical part of this, again, is that there's this, just like with all the other metabolic cycles, there are reactants that can be reused over and over again. So this acetyl-CoA is actually combining with a 4-carbon molecule. All right, we don't need to worry about the name of it just now, but that 4-carbon molecule is combining with this acetyl-CoA, and it makes this thing called citric acid. All right, citric acid is a 6-carbon molecule. So the Krebs cycle is also known as the citric acid cycle, right? Just Dr. Krebs is the guy who discovered it, so we've named it after him. The Krebs, not Krebs cycle, Krebs cycle. That citric acid, the 6-carbon molecule, goes through a series of reactions, one of which releases CO2 to the atmosphere. It also reduces NAD+. So NAD+ is reduced to NADH. Then we end up, if we lost a carbon, if we go from 6, we're down to a 5-carbon molecule. We happen to lose another CO2 in the process, another carbon. There's another reduction. So we have NAD+ to NADH and a hydrogen ion. And now we're back down to a 4-carbon molecule, which is what the rest of this cycle uses, right? We end up with another 4-carbon molecule until it binds with another acetyl-CoA. But we can't forget that there's a little bit of energy that is removed from this cycle when ADP plus P get phosphorylated to make ATP, a small amount of it. So it's really 1 ATP here per acetyl-CoA, or 2 ATP per glucose molecule. But then we also have FAD, which gets reduced to FADH2. Remember, FADH2 is going to go to make a proton gradient as well in the electron transfer chain, just like this NADH and this NADH. Okay? And then we end up with the rest of this process. So I drew this a little bigger than it needed to be, but this is going in a clockwise cycle around, and that's the end of that pathway. So what's crazy is that we've lost a bunch of carbons and oxygens. Remember that it all started with C6H12O6 in a simple way, that glucose, if glucose is the only reactant. So we've lost carbons, we've lost oxygens, and hydrogens are being sent in NADH and FADH2 along with electrons from that glucose molecule to the electron transfer chain. Some of the electrons get absorbed into this phosphorylated molecule of ATP as well. So then we go back to the electron transport chain, which is here, right? So these hydrogens end up on this intermembrane space. There's a higher concentration in this space than in the matrix, and they are going to try to diffuse, but they can't go through this phospholipid bilayer because that's a hydrophobic region. Water is a polar molecule. So hydrophobic means nonpolar, and hydrogens have a charge to them, so they cannot go through willy-nilly. They need a protein to go through. They go through ATP synthase. ATP synthase combines those hydrogen ions with the electrons from the electron transport chain and the oxygen from the atmosphere to make water, to ensure there's a low concentration gradient. And through that protein, ADP gets phosphorylated into ATP, and it makes lots and lots and lots of ATP. Okay, so that's the end of that process. I hope that's clear enough. It's a lot of detail that you certainly will need to practice over and over and over again, which I expect you to do for Friday. Okay, so I hope you're not watching this Friday morning or late Thursday night.